Welcome to the second day of the back-to-back events of the EUGLOHRIA project. For the first session, we are going to have a presentations from the research projects of the EUGLOHRIA seeding grant. They will speak to us about their best practices and lessons learned in the shared use of the research infrastructures in global health. For the first presentation, we'll have the researcher Maria José Oliveira from the University of Porto, and she is the PI of the Sensitize project. Maria José, please feel free to take over whenever you're ready.
So thank you very much, first of all. So I would like, in my name and in the name of my collaborators at the University Paris-Saclay, to acknowledge the opportunity of the rectory of the University of Porto, but also of the network EUGLOHRIA and EUGLO for the opportunity to present here our work, and above all for the opportunity for having that you have financed our project that is now starting with many interesting results, and that I hope is fostering our collaboration and research. So the project is called Sensitize. We aim to dissect the tumor microenvironment to battle cancer radioresistance and to avoid the immune escape, that cancer cells are able to escape the immune defense. So actually, I would like to show you the faces of the persons that are involved in this project. So as I told you, this is a project that is established between I3S here from the University of Porto and the Institute Gustave Roussy from the University Paris-Saclay. And particularly, we are two PIs, because this is a joint project. And from the University of Porto at I3S, it is myself, the PI. I'm a biologist and I'm working in tumor immunology. And from the University Paris-Saclay, the PI is Professor Nathalie Drouin, that she's the head of the genomics platform of Institute Gustave Roussy. So together with us, we have a team of people that are already dedicated for a long time in collaborating and working together. So Flavio Castro is the junior researcher on my group that is expert on tumor immunology. Joana Paredes is a group leader at the Institute that is an expert on breast cancer biology. And together, the three of us, with Professor Eric Dutch, that is the head of the radiotherapy department at Institute Gustave Roussy, we are co-supervising Ana Borges, that is a PhD student that is engaged in this project, and she is already a co-tutel process student between ICBAS, the University of Porto, and the University Paris-Saclay. Besides these people I present you, we also have from the University of Porto the engagement of Hugo Osorio, that is the head of the proteomic department, the proteomic facility and platform here in I3S. And from the University of Paris-Saclay, besides Professor Nathalie Drouin and Professor Eric Dutch, we have two researchers, that is Dr. Michele Mondini and Lydia Mezziani, that are experts on radiotherapy and also on tumor immunology, and that belong to the group of Professor Eric Dutch. So actually, this work has a major contribution from the genomic platform of Institute Gustave Roussy and from the University Paris-Saclay. Professor Nathalie Drouin has the platform engaged at the Institute, and the Institute is, and I think it's very important you know, is a world reference hospital and center for cancer research and treatment. The radiotherapy department is one of the best and the most engaged radiotherapy departments in the world, and they are at this platform engaged with a lot of equipment that allow us to go for whole exome sequencing, but also for single cell sequencing. So we have used the services of this platform through this project to perform some RNA detail analysis on spheroids of cancer and immune cells, as I'm going to show you. And we had not only the expertise of Professor Nathalie Drouin, but also from one engineer, that is Maela Franciletti, and the three technicians, Betty Late, Andre Manaimo, and Fatma Kensu, that are working at this facility, and that were helping us not only on the acquisition of data, but currently still in the analysis because it's a long process of work. So to tell you a little bit about the projects that we are engaging, we are mainly centered on triple negative breast cancer because triple negative breast cancer is one of the most aggressive subtypes of breast cancer. It lacks the expression of receptors like ER, progesterone receptor, or HER2 that are normally target of other therapies in breast therapy, but they are not expressed in triple negative. That's why it's called triple negative. And due to that, it escapes the conventional therapeutic approaches. It's normally very radio resistant. That is one of therapeutic possibilities. And therefore, we wanted to develop and understand better the mechanisms to be able to identify possible targets that could be combined with radiotherapy to sensitize tumors to this therapeutic strategy. And to understand this better response, we need by sure to look to the tumor microenvironment, because it's not enough to look to the cancer cells that are radioresistant, but it's also important to understand how the other cells that are present at the same environment, in the same neighborhood, may participate in these radioresistance mechanisms. And at the tumor, we have a lot normally of fibroblasts, but we have a high component of immune cells. And given our interest on tumor immunology, we are trying to understand how a particular myeloid population that are macrophages present at the tumor microenvironment might contribute to triple negative breast cancer radioresistance and immune escape. So in fact, this is very important in the case of breast cancer, but it can be applicable to other tumors. And that's why we like also very much this project, because it could give us hints to other applications for many radioresistant tumors. It is estimated that about 50% of all tumors are once irradiated, and many of them are relapsing and radioresistant. And we believe that finding these mechanisms, they might be transversal and applied to other type of tumors. So together we designed a project that aimed at first to try to develop a biomimetic 3D model that could gather together cancer cells and macrophages. We wanted to get out of the conventional monolayers or 2D cultures. We wanted to go to a 3D culture model because this would allow us to respect the structure of the tumors, but also the diffusion of nutrients and metabolites. After this, the project aimed to dissect molecular mechanisms that are underlying breast cancer radioresistance, and I will show you that more in detail. And our future aim would be that once we find these targets, we could identify inhibitors of these molecules and combine them in an animal orthotopic model with radiotherapy to confirm their ability to impair radioresistance and to sensitize cells. And the idea is then to go back to the clinical data and to compare tumors of patients that respond well and that did not respond to radiotherapy and confirm that these therapeutic targets are indeed there, differently expressed. But the EUGLOHRIA project that I'm presenting to you is just centered in these two phases. So the development of these 3D immune spheroids, their characterization, and the dissection of the underlying molecular mechanisms. So for that, I'm showing you the first results we achieved, but also the methodological approaches that we have performed. So what we have done was building up spheroid models, and this is a very simple methodology, I should say, and I think this could be of interest to many of you, because it's based on molds of agar that we can apply to different wells of different size of plates. And actually, in this case, we can afford to have in each well of a in each well of a 6-well plate, 81 microspheroids. These microspheroids are very similar to each other. They are very stable. And afterwards, you can use these spheroids in our case, to submit to treatment, and the treatment in our case is radiotherapy. So we can irradiate the spheroids directly. I'm sorry, I'm going to try to use this. So we can irradiate the spheroids afterwards, and the spheroids are developed during 3 days. They are then irradiated for additional 5 days, because we want to mimic 1 week of a patient's treatment of radiotherapy. And we are using two different doses. We are doing 5 fractions of 2.67 or 5 fractions of 5.2 Gray, because we want to mimic the long and short treatments that normally these patients may engage. So we can mimic then this 1 week. We take these spheroids directly to the radiotherapy department, where they are lying in the same table where normally a patient does radiotherapy. So we are intercalating our plates with the patients, and they are exposed to the same conditions, and then we bring them back to the lab to analyze. And what is nice is that then you can use these spheroids to dissect them, so we can dissociate the spheroids, perform flow cytometry. We can keep them and analyze by confocal microscopy, or for a different type of screening, like we have also learned and performed with a colleague here present, Andre Maia. And so we can analyze live dead, we can analyze proliferation, and we can also at the same time maintain these spheroids or sort the different populations, separate macrophages for one side, cancer cells for the other, and we can perform detailed proteomic and RNA analysis afterwards, and that's what we have done, as I will show you. So afterwards, just to go through. So we have in the first task, we have analyzed how these macrophages are. And what we could see, I will do it simply, but I think it's important that you can see that after 1 fraction, there is not much alteration on the profile of these macrophages that have been in contact with the cancer cells. But after 5 fractions, so 1 week of radiotherapy, we see an enhancement of expression of markers that are considered as pro-inflammatory, and we have a decreased expression of markers that are considered as anti-inflammatory. So this is suggesting that upon irradiation, even in the presence of the cancer cells, these macrophages are activated in a good manner. They are becoming pro-inflammatory in a way to fight and to counterbalance the tumor growth. But what we have seen in meanwhile is that these macrophages are favoring cancer radioresistance. Because if you could appreciate here, after 1 week of irradiation, if you see the control cells that are the empty bars, once you irradiate with 2.67 or 5.2 Gray, the number of live cells decrease significantly. So the cells are dying once they are irradiated. But if they are irradiated in the presence of macrophages, that are the bars with the black indication, these, as you can see, cancer cells are no longer dying. So even in the presence of macrophages that should be protecting, that have a good pro-inflammatory mode, these cancer cells are escaping death. And so they are getting radioresistance. And we could see that they are even proliferating in the presence of these macrophages. So therefore, we decided to understand which are the mechanisms behind, and this is just a short summary. So if you have tumor cells, and these tumor cells are in spheroid and they are irradiated, there is DNA damage, the cells are being destroyed and they start to die. But if they are in the presence of macrophages, the cells are saved and they are indicated to survive. And in parallel to that, we see that the macrophages become pro-inflammatory, what seems good, but cancer cell proliferation is occurring, and there are several types of cytokines that are being released that are possibly attracting other populations and other types of cells. So we tried to look this in more detail, and that's through the EUGLOHRIA project we have done it. And what we have done was to use then these spheroids that have been irradiated or not, and we have then the possibility to dissociate them from the cells that are radioresistant, the macrophages, and we can even analyze the secretome. So we have done at I3S the analysis of secretome in the multiplex ELISA array, and we have done at I3S the mass spectrometry, as I have told you, with the platform proteomic service from EUGLOHRIA, and at Gustave Roussy we have performed the RNA-seq analysis. So to briefly show you our experiment, so what we do is that we take the spheroids of cancer cells that are not irradiated or irradiated with the 2 doses, without macrophages or with macrophages, and we can take at the end of the 5 fractions, we can take this to collect medium for multiplex ELISA array. In that way, we can identify factors that are present in the medium, cytokines, chemokines that are being released. And we have also performed the detailed proteomics that I cannot show you at the moment the results because we are still ongoing with analysis. What we have then done also was to go further and take the spheroids that were performed, and we decided to sort them here at the flow cytometry platform at I3S. So what we do is that here is simple, we just have cancer cells, but here not, we have a mixed population. So we do a selection for the CD45 positive cells that are the macrophages that are here going to be selected in one tube, and the cancer cells that are CD45 negative will be selected in another tube. And then we analyze these 2 populations independently by RNA sequencing, and these allow us to identify the signature, the molecular signature of the cells that have been alone with macrophages and compared with ones that were without macrophages. And we can also compare the ones that have been irradiated in the presence or not of macrophages at different doses. So I think this can provide us a lot of information and on the molecular mechanisms. So what you can see, I just show you very briefly, not to overload you, is just these are called volcano plots, and they allow us to compare the genes that are overexpressed or downregulated in one condition to the other. So what we can see is that the cancer cells, for instance, that have been irradiated with these fractions or that fraction, we can see that there are a couple of genes that are upregulated upon irradiation and others that are downregulated. Each of these dots is one gene that we can now look in detail and see what is the gene that has been altered. And this was the same when we compared cancer cells that have been irradiated or macrophages that have been irradiated but previously in contact. And if you look just overall, you can immediately see that the cancer cells that have been in contact with macrophages, they have a minor genetic variety compared with the cancer cells that have been not with macrophages. So they have less differences and they have less genes upregulated and less genes downregulated. And you could see then we could not detect major differences genetic between the one fraction or the other fraction, showing that apparently in vitro they are working very similar. Very few genes are here altered, and no genes were differently expressed in macrophages exposed to the different radiation schemes. So when we look now, I have just the 3 details to finalize these. If we look now to the genes, we can now look to the families, to the type of genes and the families that are upregulated or downregulated. And as you can see, we could see that when we compared the cancer cells that have been irradiated with ones that have not, without macrophages, our major alterations are on DNA replication, cell cycle, DNA damage repair, and the G1 to S cell cycle control, so the cells go or not to proliferation. If we look to, by the other hand, to the cells that have been in contact with macrophages and that have been irradiated, we have an activation of the leptin pathway and immune response, IL-2 signaling, IL-4 signaling, and lipid metabolic alterations. If we look to the macrophages now that have been in contact with the different with the cancer cells, The ones that have irradiated have these couple of genes upregulated, these downregulated, and they are related to photodynamic therapy with leptin pathway, with glucocorticoids, and with DNA damage response. So now what we are currently doing is that we are trying to identify these most up-alterated genes, and that we can identify and that we can validate also in an in vivo model. So these were the major differences. I will not go through them, but just if you can see, when we have cancer cells and irradiated in the absence of macrophage, they are several types of molecules that are differently expressed, altered, upregulated or downregulated. But if you look to the sign when they have in the presence of macrophage, the majority of these molecules are saying equal, so they have no altered expression. So meaning that the macrophages are saving the genetic alterations induced by radiotherapy that normally are leading to the cell damage. So the end, what I would like to tell is what is the future? That was a question you have asked me of this initiated project. So we have several ideas. Of course, we want to keep dissecting these molecular mechanisms. We would like then to combine inhibitors of these molecules in the animal model. And we took already the opportunity because we had in parallel with EUGLOHRIA, we had a PSOA project that was allowing the interchange and mission between researchers, between Paris-Saclay and the University of Porto. And so the student Anna went 3 months abroad to Paris-Saclay, and she had then already, while doing the RNA-seq analysis, she was already starting with the development of the animal model, and she could already inoculate cancer cells in the mammary gland of the mice. She could see that the controls are developing disease, the tumor is growing. If we are irradiating, they are also growing, but there is a delay of growth given to the irradiation. So we have now the model ready, that once we have these inhibitors, we can combine in this animal model. So it was already an output of the EUGLOHRIA project that we could already optimize also the animal model. So as the end, we would like from that, as I told you, to validate at the clinical level. And for what would you need? What do we need? We need, of course, to involve, I think, this co-tutel of Anna in the EUGLO network, because she's in fact financed by the Portuguese Science Foundation, but she has indeed this co-tutel between the two universities. And we have this approved PSOA project that will allow us to keep the missions, but our priority is to have additional funding for reagents. So we would be very attentive to the initiatives of EUGLO and to novel EUGLOHRIA applications. We will be look for you, but also we are aiming to do a LACACH application that the deadline is now in 16th of November. We would like to make a cost action that will be headed by the Gustave Roussy and by Professor Eric Dutch on radio resistance. We are preparing also an FCT joint application, and we will use not next year, but we hope if we can next year apply to a cost action, we have the network established to the year after try for an European project application. So this is our wish, but let's see how we can, we have to work hard for that. But the experiences that we can come out, I thought that this was an extraordinary experience to all at professional and personal levels. It provided technical training, and for the co-tutel, a PhD student, for her, I think this was really very important, and provide training in the two senses. Porto went to Paris to establish the spheroid cultures at the laboratory of radiotherapy. They didn't have the spheroid cultures established, so Anna was there and established the spheroid cultures. And I was there last week, and I was very pleased to see that they are already at least 2 PhD students working on this approach. The Paris-Saclay, by the way, helped us a lot and instructed us with RNA-seq data acquisition and analysis. This EUGLOHRIA provided a fast access to a high throughput technique that we would not have by the cost of the technique in another way, provided access to the highest standard quality of data acquisition and analysis, reinforced the collaboration we had and appointed novel collaborative pathways, and provided the possibility to obtain a robust preliminary data that would, I hope, strengthen further applications. When you ask about the added value of using the platforms, this is by sure, but I can tell you that either here, I think the scientific platforms of I3S are really one of the most precious infrastructures we do have. But also there, the scientific platforms are very important because they standardize equipment and methodology, minimizing optimizations. There is a real, true scientific and technical expertise fostering research, is development of novel ideas and projects, promotes scientific discussion and scientific advances, and at the same time there is an intercultural, academic, scientific and technical interface. So I just have in my name and the name of all team from University of Porto and Paris-Saclay to acknowledge you for this opportunity. So thank you.
I don't know if you have any questions for Maria.
Thank you very much, Maria José. We'll now move forward to the presentation of Axel Imhoff, the PI of the very sorry.
Thank you very much. It's not a question, this is a remark, a comment. That's fantastic because you have performed a nice work. Perhaps you had already a collaboration between your team and the team of Eric Dutch, etc. But you have relied on EUGLOHRIA. This is only a comment. How can we help you for the future steps?
It's very good. I was thinking on that, and I think indeed, for instance, one point that for me would be very nice at human resource level would be that I would like very much that Anna, because when she applied for FCT, these grants of EUGLO were not yet available, so she got the FCT grant. But I would like very much that she could be engaged through this co-tutela to the EUGLO network, and that she could also benefit from some opportunities that would maximize this interface. We have these missions on through the project PSOA that are just finishing at the end of the year. So Professor Eric Dutch is coming to visit us now at the 7th of December. And then afterwards our missions are finishing. So if there is opportunities also to support these missions that are very important for our interface and at the same time for bringing samples to, at the same time we are trying to do meetings. For instance, normally when Professor Eric Dutch comes here or I go there, there are open sessions through the Institute. We went with Professor Eric Dutch to visit the Oncology Institute, in particularly the radiotherapy department, and this brought that there was a clinician doing her specialization at radiotherapy department. She's currently at Gustave Roussy, so I think this is bringing the possibility of people to interchange. So I think these missions are also very important. And then we have the sides of the financing. So this is something that for us would be also very important. Would be also important that we could get some regular or the type of funding like this opportunity that was provided now. To give you an idea, I can be very honest to you, the value of the platform that was paid was half of the real cost of it. So it was directly the Professor Nathalie Drion. She said the project is very interesting. We are going to cover it, but there was just half of the cost that we could, because otherwise we could not afford it. It was over 10,000 euros, so we could not afford it. So I think this is by sure, if you have these opportunities also to feed these type of projects, in my case and the case of the colleagues, I think these are very important because in our case allow us to do technique that we were not able to do it, brought us preliminary results that we can use to apply to other type of grants, and it would be very nice if through you, and I think through EUGLOHRIA, I think through these networks we can get more in contact with other colleagues that are working in this field, that we can make an interface, that we can create a network, scientific criticism, because to me there is, of course I'm very much focused on the field of radio resistance, but resistance in cancer overall, not not only radio, is a major problem. It's that is killing patients. And so we need to understand resistance, and we need to make big networks of people, multidisciplinary approaches that can look to it in a different way. So bringing people from different universities, EUGLOHRIA, I think, would help us. Thank you.
Thank you very much for this presentation. I'd just like to underline the interdisciplinarity of your project. I think, I know Nathalie Drion, she works with Hugues Talbot. She is responsible for the data science for health at Paris-Saclay. So I think it's very interesting to collaborate with other teams in the problem of personalized radiotherapy. With data science, and I think that laboratory like Listen or Mix will be very interested in collaborating. So I think that it's the starting point of new collaborations inside the EUGLO.
Thank you very much for your comment, because actually, so we had a major help of optimizing everything and also the data acquisition, but now we are dealing with this very effort on data analysis, and they are helping us, but they have also, through the collaboration with other colleagues at Gustavo Rossi, trying to bring this big data analysis network that we can improve this analysis, because it's very easy to make a mistake. So what we want to do is to be with the experts that are guiding us and saying, no, you should not analyze this way, you have to analyze this way, that we can really have candidates that will be then, of course, they need to be validated, but that we could come something more reliable at the end and close to what is going to be relevant in the clinical setting. But thank you very much for your approach. I will probably ask you the names and that we can speak. That will be very precious for us. Thank you very much. Thank you. Please.
First part of your presentation,
Yeah,
but
no problem.
did you check the ratio of the M1, M2 type macrophages before and after irradiation?
Yes.
This is my first question.
Yes.
The other one is a technical question, that how deep can you screen the living 3D culture with any kind of techniques? Thank you.
Yes, so regarding the first question, yes, we did. So what we normally do, so what we would like, I will tell you that we would like very much to have done this approach with the cancer cells and macrophages derived from patients. But as you can imagine, this is so many cells that we need that we could not afford to do that, because from a cancer patient, we would have a maximum 4 million of monocytes, and here we needed over almost 100 million. So we could not afford to do from that. So our idea is that we started from these cultures with the cancer cells and healthy monocytes derived from healthy blood donors. And that once we dissect the mechanisms, we can go back to the patients, and then we know what we have to look, and we do in a minor scale, and we try to see if they are those. For that, we have also looked to the proportion of macrophages. So normally we started within our culture, because this is breast, we started at the beginning with 60% of monocytes versus 40% of cancer cells. And at the 8th day, if we don't irradiate, we have 20% of macrophages and 80% of cancer cells, because they are proliferating and the macrophage population is then proportionally reduction. When we look to the profile, the M1, the profile of these macrophages, they have a mixed, because they are in the presence of cancer cells, they have a mixed phenotype. What we have is over 40% of expression of anti-inflammatory markers, CD163 and CD206. And once we irradiate in co-culture, the 206 drops very much. And we have a high increase of CD86 and a high increase of HLA-DR. So the pro-inflammatory are raising up. So we are, even in the presence of cancer cells, if we irradiate, we are able to convert these macrophages in a good manner, pro-inflammatory. And from previous work, we know that is because they are not dying to radiotherapy and they are activating pro-survival pathways via NF-κB that induce the release of interferon gamma. So that was, I'm sorry, more technical part. The other question you have asked was about how we do it. So we do the co-culture. After the 8 days after the irradiation, we do sorting. So we label the cells with an anti-CD45 antibody, and we sort for CD45 positive and CD45 negative. So the myeloid cells, macrophages, will be the CD45 positive, and the cancer cells the negative. And then we take these isolated cells and we do on them proteomic and RNA. I don't know if I answered your question, but otherwise we can also speak afterwards. Okay?
Yes, thank you very much, Professor Maria José. We're going to move forward now with the presentation from Axel Himhoff. He's the PI of the PIPA project. So please, when you're ready, Axel, take over.
Yeah, good morning, everybody. I hope you can hear me.
Yes, we can hear you fine.
Okay, good. So I have to apologize for not being there. This is also because, can you see my presentation now? Can somebody comment on that?
No, we saw it.
Yes.
Now it's not there anymore.
Oh, okay.
It's there.
We can see it.
Yeah, it's there. It's there. Perfect.
Okay, good. So this is somewhat different from the impressive presentation that you just heard, because this is an entirely novel project that was initiated thanks to a meeting that we had within the EUGLOHRIA network, where all the platforms came together that work on proteomics. And what we decided that would be good within EUGLOHRIA to establish interactions and collaborations between those platforms that would then allow us to further establish best practice methods and methods on how to analyze things. And so in the project that I'm going to present to you today, we teamed up with the platform at the University Paris-Saclay to investigate best practice methods to automatize and very deeply and very precisely investigate pathogens derived human proteins in human plasma and saliva. And we wanted to use as an example the SARS-CoV-2 as kind of a prime example of pathogens that we may be able to detect. So this was called, as I said, this was a collaboration between the proteomic platform at University of Paris-Saclay, which I learned during our first mission is that our first meeting is actually not in Paris, but in Avignon. So because Paris-Saclay is a big consortium of many platforms. This was a very nice interaction and continuous interaction that we had with Lucia Grange, who's actually heading this platform, or co-heading this platform. It's a very impressive platform with 5 high-resolution tandem LC-MS systems, where they use actually explore, the state-of-the-art mass spectrometers. I won't go into details. Their mission is clearly to look at proteomic compositions, especially in light of pathogens, and this is something which made the collaboration, or makes the collaboration very interesting for us as well, because they have labs in Avignon where they can grow and propagate even highly pathogenic highly human pathogenic viruses, and that would then allow, which we don't have in Munich, and that would allow us to collaborate with them and look at and fulfill the question that we wanted to ask. So we in Munich are also a protein analysis platform that exists since 2002, was initiated by a couple of German research networks. We have 7 LC-MS mass spectrometers, very similar to what the colleagues from Paris-Saclay have. In addition to that, we have 2 MALDI-TOF instruments that we use for MALDI imaging and for just regular analysis of peptides. And we serve a couple of labs in the medical faculty in the Munich area. So what did we want to do precisely? We wanted to apply the mass spectrometry-based methods to comprehensively analyze pathogens in human biospecimens. So this is a very broad question. The EUGLOHRIA project was, the idea of the EUGLOHRIA project when we applied for it was to just establish a collaboration and an interaction between the two labs. And I think that worked fantastically. And there was a lot of exchange in that with regards to methods and information, so that in the future we can actually use those optimized sample preparation and optimized protein analysis to get a deeper coverage of pathogen-derived protein. And then hopefully, and this is all, I'm projecting this in the future, as I said, this is was not an existing collaboration, that in the future we can validate and investigate pathogens. And initially we're focusing on viral proteins, but of course that could be expanded on other human pathogens like bacteria or fungi, using either targeted or untargeted proteomics. What were we hoping? And I must tell you, we have established an interaction, but I can only tell you the expected outcomes because we have very limited results at the moment. The idea was that, of course, we could use quick and fast and cost-effective mass spectrometry to quantify human pathogens, and we were using SARS-CoV-2 as a proof of principle. And we wanted to get a deeper insight then potentially on the mechanisms of pathogen infections and how that might actually affect host physiology, to get an idea on how to develop alternative strategies to fight the human pathogens. And most and foremost, and this is what we have done in the project so far, is to exchange best practices and exchange approaches between the collaborating team to allow the proteomic platforms within the EUGLOHRIA network to interact with each other, talk with each other, exchange ideas already at the level of the postdocs and PhD students to get the best tips and the best approaches to potentially fight future pandemics, if and I'm sure there will be, with a more efficient way and a more collaborative way. So what have we done so far in Lucia Grange's team? As I said, what they have, and I hope you can actually see my pointers. What they have is they have grown SARS-CoV-2, and they could then add virus particle to human saliva at various concentrations, and then analyze of how many particles would one need in order to detect the individual peptides. And they just kind of quantified this, and the virus load was actually quite high that you had to do. Plus, they were actually looking at very targeted peptides that are present in all, as you can tell, I'm affected by this at the moment, that there are can be present and found in all strains of that. So what we did together with the samples that we got from Paris-Saclay and what we had established in our lab, we wanted to do an untargeted high throughput shotgun analysis where we can then look at all the peptides rather than only a few individuals in a rather fast mission, and then do this in an automated way so that we can get large sample numbers, ideally in 96 well plates or higher amounts, and then automate in an automated fashion analysis. And to do this, of course, you need to avoid pipetting or have a system where you can do this all by robots. And we use, that's now very technical, SP3 single pot sample preparation, where we just add a protein mixture. And of course, currently we're doing with all kind of with all kind of human specimens, but also with the specimens that we got from Claire, to just enrich the proteins and ideally all the proteins from this protein solution that we have, then digest them on beads, because this is how you analyze proteomic measures, and then take the supernatant and analyze it by LCMS system. This entire thing is established on a robotic platform which processes 96 well plates in about 4 hours. So this is the throughput of sample preparation, and we're currently optimizing this for the SARS-CoV-2 methods. Those samples are then loaded on what's called EVO tips, which are just kind of trap columns on the for the proteomic measurements, which can then be automated can then be taken by another robot to feed them in a mass spectrometer. And we use data independent acquisition, which is a very efficient way to do shotgun proteomics, so that we can currently do in a routine way 60 samples per day, but we're quickly approaching throughput of about 200 samples per day, which was greatly turn over the throughput that we can have, which is basically just limited by the speed of the mass spectrometers. Of course, if you want to ever put this in a setting where it can be used in a clinical system, where the samples come from the patients and then they have to be analyzed rather quickly, and especially also compared among each other, it needs to be very stable. And so this is what we've done over the last couple of months, where we just did large sample series, and this example here is a sample series of about 4,000 plasma samples, and every 12th sample we added a quality control standard. And I hope what you can see is that over time, if we look at the identification of the proteins, either in total or in individual proteins, so each line is a given protein, we actually get quite nicely reproducible data, so that we could envision that we can take the samples even from patients and look very stably for pathogens. As I said, we've just establishing this, and we're in the process of having everything done, and we got the samples shipped. We bought the consumables for it and have actually started to load it. Currently, the samples are running. The same is true for in Paris. What we also wanted to do is to do this a bit more quantitatively and use isotope labeled standards. This is something we haven't done because we have to, in the shotgun approach, still look at the peptides that we can efficiently analyze this. So what I wanted to show you here is how does EUGLOHRIA add to this interaction. One thing that I already mentioned several times here is that clearly without EUGLOHRIA and without the meetings that were initiated by EUGLOHRIA, this particular interaction and this particular project wouldn't have started. We wouldn't have done those experiments. And I, and as well as Lucia, asked their students and collaborators of what they think about the EUGLOHRIA project. And those are just quotes that the people in Paris-Saclay very much like to interact and collaborate with us. And we, of course, do this as well. So this is Teresa Barth, a postdoc in my lab that is primarily working on clinical samples. And of course, she's very excited to have the opportunity to look at human pathogens in a defined system and look at the possibility to automatize it and get information on how mass spectrometry and platforms can be used for efficient and fast diagnosis. This takes me already to the end of the presentation, so it's a bit short, but I hope I gave you a little bit of an insight of what we are currently doing and what we're planning to do. I'm very thankful for EUGLOHRIA to have made this possible and to initiate this, and of course the money we get is only to start the interactions, but of course we're planning to apply for further research money to extend this collaboration and then help us to do this. So as I'm not there, unfortunately, I cannot offer you to chat in a coffee break or something. So if you are interested in the project, if you want to chat with us, get in touch. So those are the websites, but you can also download or get information from the EUGLOHRIA website, where all our emails address. So drop us a line, either me or Lucia, if you like the project or if you want to collaborate in any way or form. We're more than willing to help, not only on pathogenic substances, but also on proteomic analysis. So with that, I'd like to thank all of you who listened. And if you have questions, just tell me now or drop me a line via email. Thank you.
Thank you very much. Thank you very much, Axel. We're just checking if there's any questions from the public. Okay, I think not. Thank you very much for your presentation. It was very insightful. We're going to move forward to Judith Dennis, the PI of the PRINCE project. Judith, please take over whenever you're ready.
Hello and good morning to everyone. Can you hear me and see my presentation?
Yes.
Yes.
Yeah, okay. So thank you. So first of all, I'm sorry that I couldn't go to Porto. It would have been very nice to be there, but I will try to show my presentation through this online possibility. So thank you for that. So our EUGLOHRIA project is a collaboration between the University Paris-Saclay and the University of Szeged on identification of ligands for a long non-coding RNA, which we thought to be a good target to use as a proof of concept for RNA targeting. Okay. So the two PIs we work together are me from the University of Szeged and our Professor Jean-Christophe Sintra from the University Paris-Saclay and the CIA laboratory in Saclay. So he's a medical chemist by training, and he had his PhD also in this field and his habilitation. He is the head of the laboratory for bioorganic chemistry, and he has several interests in terms of translational research and finding small molecules as inhibitors for biological processes, which could be used or later on as drugs or medicals. And he also has a number of patents connected to this. He's also the leader of the high throughput screening platform at Paris-Saclay, which the CAPS platform, which allows to the screening of numerous chemical molecules, small molecules in various settings. So this is where we started to collaborate. I am a molecular biologist by training and got my PhD at the University of Szeged in immunology. And since finishing my PhD, I still work at the University of Szeged from the last year also as an assistant professor. This collaboration came through the EUGLOHRIA platform for us. So let's go for the project. So what we wanted to do was to identify small molecular ligands which might bind to the PRINCE non-coding RNA and alter its functions. So our research on the molecular basis or the biological basis comes from keratinocytes. These are the cells which build the epidermis, the outermost layer of the skin. And for decades, it was so that they are just structural elements of the skin, but actually they have a lot of immune functions, as we and others have found. And what we try to do is to find these immune functions and find what these cells are capable to. So originally, Prince was found in keratinocytes in psoriasis, and later on, we started to work on the keratinocytes to see how their activation through their pattern recognition receptors led to the production of inflammatory mediators, and their inflammatory mediators also activate other immune cells, professional immune cells, and recruit them back to the tissue. And one point is signal transduction, which happens after the receptors are activated. This is quite well covered in literature as well, also for a lot of cell types, also for keratinocytes. And which is also quite well covered are post-translational modifications like phosphorylations, inflammasome activations, ubiquitination, which can alter the function of the produced mediators. However, what we became interested in it in SAGAD were post-transcriptional modifications. So this is the question, what happens with the mRNA? So of course, mRNA can be translated, can go through splicing with functional consequences, and non-coding RNAs can also lead to functional consequences, can lead to the stabilization or degradation of mRNAs, and this is where our interest came in. So what happens between the transcription and the translation in this post-transcriptional phase? When we speak about RNAs, the most common types are the messenger RNAs, which encode for protein. However, there are a lot of RNAs which do not code for protein. These are the non-coding RNAs. And of them, there are a lot of housekeeping RNAs like tRNA or ribosomal RNA, or SNN SNO RNA, which have a function in the nuclei. But we have also regulatory RNAs like the microRNAs and siRNAs, which are quite fairly also studied. And we have long non-coding RNAs, which are just as long and usually have a same structure as an mRNA. However, they have no protein coding ability, they have no open reading frame, and they will execute their effects not through translation, rather through binding to their targets in RNA form. So what we my research group, my research group at University of Szeged and my PhD was based on this Prince non-coding RNA, which is a psoriasis-related non-coding RNA induced by stress. And what my colleagues have found almost 20 years ago was that this non-coding RNA was very highly expressed in psoriatic patients' skin samples, but only in the skin samples of psoriatic patients which had no symptoms. So these were non-lesional skin samples. And when we compare it to healthy or to lesional, the expression was really, really high in this non-lesional epidermis. So we thought it was thought that this could have an effect on the susceptibility for psoriasis. Later on, it was found that cellular stress would induce its expression, and it also regulates cell viability during the stress exposure, and can regulate the expression of some anti-apoptotic molecules and interact with proteins and microRNAs, and its expression is also regulated by inflammatory signals. So this is where I came in. And during my PhD project, we found that if we have prints in a high amount, similarly what we also see in the non-lesional skin, then the expression of several inflammatory cytokines, inflammatory mediators, will go down, such as IL-6, IL-8, IL-23. However, others will stay the same, which suggested that it's a tightly regulated process, how prints can decrease this expression. Firstly, we were focusing on IL-6 because this was the most robust target. And by doing some in silico biological tools, we found that the IL-6 mRNA had a potential binding site to the Prince RNA, and this could show how Prince was able to regulate its expression. It was also described at that time in a paper that the CCL5 mRNA might also bind to Prince through a distinct binding site, as we found by the bioinformatic tools. And indeed, when we looked for expression of IL-6 or CCL5, we both found that Prince overexpression would decrease both IL-6 and CCL5. However, if we disturbed this binding site on the Prince non-coding RNA, what we call the delta Prince, then the effect for IL-6 would diminish. However, for CCL5, which binding site was not affected in the delta Prince, the effect would stay the same. So this was our initiative, or like all the results, which on which we started this project. And we wanted to set up a high throughput screening a method for the identification of molecules targeting Prince. And targeting Prince is a therapeutic tool to stabilize its expression or stabilize its binding to IL-6, leading to IL-6 decrease in the case of inflammation and leading to the amelioration of inflammations. So we wanted to use IL-6 and Prince interaction as a proof of concept and develop a cell-based assay, which then we could transpose to the 96 well high throughput screening format. So for that, we decided to use this PMER-GLOW based study. So PMER-GLOW vector is usually used for the study of microRNAs and to find the targets of microRNAs. In this, but there were some publications which also claimed to work with long non-coding RNA binding sites. So we decided to try this approach. So in this plasmid, we have a firefly luciferase gene, and in the 3' UTR of this luciferase gene, we can incorporate the binding site from the mRNA. And the idea is, if there is a binding to, so after a transcription of from the plasmid, if there is a binding to the mRNA of the luciferase, then the luciferase protein will not be translated, will be degraded, and there will be no luciferase activation. If there is no binding between the two partners, then we will see light emission and activation of the luciferase protein. So the first step was to get this plasmid and clone the mRNA binding sequences to this 3' UTR of the luciferase gene. So we had 5 different plasmids for this: the whole IL-6 binding sequence, fragment of the IL-6 binding sequence, the CCL5 binding sequence, and we also had some results on IL-23. a cytokine also involved in psoriasis pathogenesis, suggesting that there could be a binding. So we also included this one. So this is the setup, how it looked. So we had 5 different plasmids, and we had a Prince overexpressive plasmid, and we co-transfected these plasmids into the cells. We used also a keratinocyte cell line for this, the HPV CAR cell line. And so we co-transfected this and waited for 24 hours. And we thought that if binding occurs between the Prince and the luciferase, then we would see a degradation and maybe no light or less light emission in the end. But if there is no binding, then the luciferase can be translated. And then by adding the luciferin to the system, we can measure light emission. There are commercially available kits for this. We thought that this would be a good point or starting point, because luciferase activity measurement can be used for the high throughput screening. So in our results, we saw that there was a slight decrease for the IL-6 binding site and the CCL5 binding site when we overexpress Prince compared to overexpressing an empty vector. However, there was quite a big standard error and difference between the experiments. And we also incorporated in the experiments, not just Prince overexpression, but delta Prince overexpression. So this is a plasmid where the IL-6 binding site is missing, or yes, it's missing. But we again did not see different results from overexpressing Prince or delta Prince. So this suggested, or this shows that this approach might not work in our system. We tried to optimize the transfection method, the timing of the experiments. We did quite a few experiments on this, but we didn't get any different results. So actually, we concluded that this approach wasn't really working for us, even though we did quite a few preliminary experiments. So this was not really something we can use for the HDS or transpose for that. But we were looking for other possibilities for collaboration between our groups, because we saw that this could be a very good opportunity. So we found another common interest, rare diseases, as other targets, because I am also working very closely with the group at the Department of Medical Genetics here in University of Szeged. And rare diseases are quite rare, so it affects less than 1 in 2,000 people in a population. But there are over 7,000 rare diseases, and these diseases are usually genetically inherited. And there are some diseases which are a little bit more affecting more of the population. Some diseases are really 5, 6, 10 patients in the whole world. And we have quite a few data on SILT cutaneous syndrome, which is caused by mutations to the SILT gene. And this leads to altered signaling in the skin, and leads to tumor formations with these patients having the mutations. There are over 100 mutations already described causing SILT cutaneous syndrome. However, the functional consequence of these mutations is only described for 2 of these mutations so far. So we already started functional investigations on this on the affected pathway, and we discussed this with Jean-Christophe Central's team, that by using functional studies, we could find the mutations which could be targeted by chemical molecules, and with the long-term goal for drug proposing maybe for this disease, which is a very novel way to try to treat these genetically inherited diseases, and go into this direction with a more translational research, not just functionally finding the mutations and describing those. So our future funding plans are to get a research grant from the French embassy for travel and for exchange. This is the Balaton project what we have found. This is between France and Hungary. And we would also like to start a PhD student on the topic with co-supervising between the University Szeged and the University Paris-Saclay, where it seems that there will be an international co-tutel funding program coming from next year. So we hope to go with that. So as far for our experiences, so I'm pretty sure without the EUGLOHRIA platform, we would have never met, because this without this we wouldn't have found each other, and it was a very good opportunity to travel and meet other researchers also at each other's faculty, and maybe find also some other common interests, not just the ones we already started the collaboration on. And I think it's very important to have this ongoing scientific discussion on the projects, and not just in your own team, but also with other team members or other teams, other researchers who might have a different background and might see some different inputs on your topic. For me, it was very interesting to experience a chemistry laboratory after, I don't know, 10 years without this, since I finished university, and getting to know about the high throughput screening, which might never cross my mind as a molecular biologist from translational research, and also the possibility to put our research in a more translational background, and maybe on long term find something, some drugs or some medicines which could really help people. Probably without this, I would never really come to this part of research. For Jean-Christophe, he was very happy to also connect with clinicians, since through this platform we could also, when he was visiting, he could also talk to other departments and also other clinics, and talk with clinicians. And he was also saying that, I will come to this also, that it is also crossed his mind how to maintain this network on long term. Maybe if there will be dedicated calls between the universities for long term maintaining long-term funding and novel applications. So what we think of the added value, it's so, as I already said, it's a very good platform to connect with other researchers, also on the web page, and find new collaboration, new techniques which might never cross our mind or are not available in our own institute. And it was very nice to get the funding for travel and also for research, and to have the possibility to promote our research through the EUGLOHRIA talks videos, and promote the research and the project. What we felt could be a little bit improved was the call for applications, which was not, or at least we both felt that it wasn't advertised enough. So we both got notice of it just because we know some people at the university who were involved in the EUGLOHRIA network already, so they knew it and they were talking about that. But for overall, the university was, at least in the beginning, not really promoting it to a wider audience, or like we felt it that. And also for spending the money, it was initially hard. We figured it out in the end, but for quite a while, nobody knew how to spend the money we were given. What we would also like to see in the EUGLO or the EUGLOHRIA Alliance would be a good joint PhD program or joint master's program with funding not only for travel, but also maybe for research projects or for salary of the students, so that a long-term collaboration would be really possible without many more additional funding or trying to find funding from other projects. So I think that's what we were thinking to tell. Thank you for your attention. And you can find us at our email if you are interested in projects. And thank you for your attention.
Thank you so much, Judith, for your presentation. We're just checking the room to see and online if there is any questions. We have one question, so we'll pass the microphone to the person.
Thanks a lot, Eric Casson from Paris-Saclay. Thanks a lot for the presentation. I've met directly Jean-Christophe because he was also part of the virtual tours. So we have made a movie with the colleagues of LMU. They have prepared a movie on the platform of Jean-Christophe, following the record that we have made at Paris-Saclay University. So thanks a lot for the presentation. I'm very glad to see that indeed the collaboration is progressing. Okay, we do agree with the fast process. From an administrative point of view, it was also very tough for us, very difficult.
Yes, I think we were also one of the first ones to receive this seeding grant funding. So.
Okay, perfect. You have received twice.
We had to learn, all of us.
Okay. No, one comment for you, perhaps for everyone. I think that now in the next steps, indeed, the sustainability, because what you have shown here is a proof of concept, a kind of POC. In the long term, we need to have sustainable fundings, but I think that we cannot circumvent the need to apply to competitive courses. So, for example, regarding your final remark regarding the PhD candidates, the salaries, etc., I think that in your topic, in a set of topics around that domain, we have to apply, let's say, to doctoral networks of the Horizon Europe work program. I think we have no other choice because the amount of money needed to fund a PhD salary along 3 years, typically in France, this is 120,000 euros. It depends on the countries in Europe. It's much more expensive in Sweden. It's less expensive in other countries. But anyway, if we scale, let's say, for 10 PhD candidates among the program, it would represent a large amount of money. So only the doctoral networks of the Horizon Europe work program may fund that level of fundings. Okay. But we agree that it would be a very great opportunity if we could get such a network. So we need to apply.
Yes, thank you. Thank you for your comments. I agree, we have to look also outside of the EUGLOHRIA, so it's a very good starting point.
Yes.
Thank you.
Thank you so much, Judith, for your presentation. We'll now move forward to the next presentation. We are going to hear from Anne-Sophie Bignot, PI of the Imminent Projects. Whenever you're ready, Sophie.
Hi, can you hear me?
Yes.
And can you also see my screen?
Yes, we can see your screen. It's in presentation mode, if you can switch.
So, sorry, let me switch then.
You need to switch the screen, I think.
Yeah, no worry. Okay. Is it better now?
We're seeing the whiteboard.
Okay. Give me 2 seconds. I'm more used to Teams than Zoom, actually, that's why.
Don't worry. Can you maybe also switch on your video?
Yes. Hi, everyone. So, and now, how is it?
It's perfect. Thank you so much.
Okay,
You can
so
start.
sorry for the delay. So I'm happy to be here to share with you the so-called imminent project. And so I thank the organizers for the invitation. I feel sorry. I would have preferred to be in Porto with you, but hopefully with this hybrid format and virtual presentation, we can at least share our progresses. So imminent is the nickname for immunological mechanisms of a malaria candidate vaccine to explore the transcriptomic profiling of some vaccine candidates. But first of all, let me introduce the people involved in this project. So it's a collaborative project between the University Paris-Saclay, where I am, and with the University of Porto. And so we work together, Anne-Sophie Beignon, I am a group leader at IDMIT, and Joanna Tavares, who is also a group leader on host parasite interactions at I3S at the University of Porto. And we are very lucky because we share the first EUGLO PhD student. Her name is Sonia Guerra, and that's a co-supervised and a co-tutel PhD co-funded by EUGLO through the French Agency of Research for the French part and by the Portuguese FCT. And we also have a PESOA program from Campus France and FCT for traveling and short stays. And so you can see here Sonia and Joanna and Sonia. Sonia is nowadays in Porto. And so our collaborative project aims to contribute to the development of malaria vaccines. So now I switch to the presentation of the project itself. So malaria is a mosquito-borne disease, which is caused by Plasmodium species, like Plasmodium falciparum. And you can see that in 2021, there were more than 200 million cases globally, with 600,000 deaths. Plasmodium falciparum is responsible for more than 90% of the world's malaria mortality. And you can see here the endemic region for malaria. And of course, there are different preventive strategies available, and vaccines under development can target different phases of the complex life cycle of the parasite with some liver stage and a blood stage after a mosquito bite. So there are two vaccines currently approved and recommended by WHO to prevent both clinical and severe malaria in children. Both vaccines are based on Plasmodium falciparum CSP. CSP stands for circumsporozoite protein. That's the most abundant and immunodominant antigen expressed on the surface of sporozoites, which is the form of the parasite inoculated into the skin after mosquito bite. And both vaccines, the RTSS AS01 versus R21 matrix M, differ by the number of CSP present on the VLP based on the hepatitis B surface antigen. And they also differ by the presence of or not of VLP composed of hepatitis B surface antigen alone. They also differ by their adjuvant. RTSS use AS01, while R21 use matrix M. Both provide protection, with R21 afforded a stronger protection than RTSS, but after 4 doses of vaccination and with a protective efficacy that does not last long. This is why there is still many candidate vaccines under development to perform better than the currently available vaccines. Previously, Joanna Tavares worked on malaria vaccine development, and using the full-length CSP, the immunodominant antigen from sporozoite, she could show using poly-IC that mice model, mice were protected sterilely against infected mosquito bites or against malaria sporozoite skin inoculation, but not against intravenous sporozoites challenge. And this protection was mediated by cytotoxic anti-CSP antibodies. More recently, she could also report that not only poly-IC, which is a toll-like receptor 3 and a RIG-I MDA5 agonist, but also 5' 3-phosphate double-stranded RNA, which is a RIG-I agonist, both could provide this sterile protection, in contrast to poly-AU, which is a toll-like receptor 3 only agonist, suggesting that the adjuvant play a key role in protection against malaria. And actually, that's the goals of our imminent project, to characterize more deeply the early innate inflammatory responses that are induced by poly-AU, poly-IC, and 5' 3-phosphate double-stranded RNA as adjuvant of CSP, using two relevant and complementary preclinical models of malaria. Using here at IDMIT University Paris-Saclay studies with non-human primate cells, and using at the University of Porto mice for in vivo studies. And in both complementary preclinical models, we combine an analysis of cytokine production, of cell population abundance and activation, and of gene expression profiling. An additional goal of the imminent project is to explore the capacity of vaccines to induce trained immunity. So you might not be familiar with this new concept describing the fact that innate cells like adaptive cells like T or B lymphocytes can display some memory features. And it is represented here with the canonical inducer of trained immunity, which is BCG, the current MTB vaccine. After a first stimulus and a resting phase, innate cells have been trained and they respond strongly to a second stimulus, which can be similar or not related. And this innate immune memory relies on different mechanisms than the classical adaptive memory and involves epigenetic reprogramming either of the innate cells locally and or of their hematopoietic progenitor in the bone marrow. And at IDMIT, that's one of the topic of my research, to study what kind of vaccines or what kind of adjuvant can imprint innate cells in the long term. So for the imminent project, what we do is that we compare and contrast poly-AU, poly-IC, and 3P RNA adjuvanted CSP for their capacity to train monocyte in vitro. So we stimulate cells in vitro, then we wash out the stimulus and let them rest, and we re-stimulate cells 6 days later with an unrelated stimulus, for instance, LPS, and we measure the capacity of cells to produce cytokines or to rewire their metabolisms by measuring lactate quantification, and we check if they behave differently than naive cells that were not stimulated initially. So what are our first results? We first actually tested several doses and stimulation duration in vitro using macaque leukocytes, and we compared the different adjuvant given. Remember that only 2 of them provide protection than poly-IU, although it is immunogenic, does not protect mice against sporozoite challenge. And you can see that poly-IC and 3P RNA are quite comparable for the capacity to stimulate TNF-alpha or IL-8 production by cells. So cell pellets were prepared in parallel for both cytokine and transcriptomic profiling. The cDNA libraries were of high quality, and based on the FESQ files, the RNA-seq data are also of high quality. So we are now involved in the downstream analysis of this RNA-seq data from ex vivo stimulated macaque cells with the classical alignment of the reads to the reference cynomolgus macaque genome, gene expression quantification, differential gene expression profiling, and functional enrichment analysis. So this is what is ongoing at IDMIT, Université Paris-Saclay. And after this first phase in France, Sonia Guerra, our shared PhD student, moved back to Porto to study our vaccine candidates in mice. So after a first pilot study that you can see here, just for her to get familiar with the mice model after working with macaque cells in vivo, you can see here how the 5' triphosphate double-stranded RNA together with the CSP from Plasmodium ULI provided protection against parasitemia as compared to the adjuvant alone. So based on this mice data and previous work, a new mice immunogenicity and efficacy study has been designed to compare the different adjuvants for the transcriptomic profiling of cells from immunized mice, both from the site of injection and spleen. So this different data set will be complementary. As for the capacity of the 3P RNA and poly-IC and poly-AU to train monocyte in vitro, this is a work in progress. So I'm sharing preliminary data suggesting that the dose is quite key to induce trained immunity. And for 5' triphosphate double strand in RNA, low dose were more potent to train monocyte than a higher dose. But this need to be repeated, and a larger range of doses need to be tested. As part of the EUGLOHRIA seeding grant funding, we also thought with Joanna that we should organize a workshop just to get to know each other within the alliance of university and to foster collaboration. So we will organize in November, more precisely on the 28th of November, an hybrid workshop on the mechanisms of action and pathogenesis to vaccines development in malaria. And the idea is really to foster collaborations. There will be several presentations by speakers from different universities on the molecular and cellular mechanisms of infection by Plasmodium parasites to pathogenesis with presentation by clinicians and presentations also on responses to malaria and on vaccine development. And the idea is really to highlight the high variety of research on malaria among researchers from the Alliance, and the diversity of models of studies, of tools. And so hopefully this will highlight complementary approaches and opportunities to share protocols or regions or share students and ultimately to collaborate. As for the budget, funding was really instrumental to profile gene expression from both macaque cells and immunized mice, but it also includes platform fees to study in depth cells using a mass cytometry, and to profile also in a multiplex way the cytokines that are produced in vitro with macaque cells and in vivo in immunized mice. And that was also helpful for the workshop organization. So with Joanna, we really think that this EUGLO funding for us is a rich experience, because we do share complementary and synergistic knowledge and tools to address a question that is new for both of us and which is really concrete about the immunological mechanisms that are in play to orchestrate protective immunity. But we must admit that this was obviously facilitated to share this knowledge and tools and to collaborate. This was certainly facilitated by the fact that we knew each other before. And I think we need to underline that it's also easier, likely, because we share a PhD student. And Sonia plays really a key role in linking our two teams, and she ensures the technical and scientific continuity of the project between University Paris-Saclay and University of Porto. As for the added value of the platform, again, it allows us to address the same question, but using two models. And this will ultimately converge towards a preclinical study in vivo in non-human primate. And this is a key step for vaccine development with a clinical trial ultimately, of course. As for the added value of EUGLOHRIA, this is an alliance of European universities, and I think it's important to act locally at the scale of Europe and to tackle global health challenges, not only by combining education and training, but also research, which is more new. And this funding allowed us to perform a transcriptomic analysis to discover what differs between protective and non-protective vaccines differing by their adjuvant. And I think we will learn, of course, for the malaria vaccine development, but this knowledge will also contribute to other vaccines, actually, that might use these adjuvant. And this seeding grant was really a key to strengthen our data and our project and our collaboration. And the goal now is to submit proposals to the French Agency of Research and to EU, of course, to get more money to do more ambitious project. But that was a good start. So I thank you for your attention. Of course, above all, I thank Sonia Guerra, our shared PhD student, EUGLO PhD student, and Johanna Tavares, but also Eric Cassan from EUGLO University Paris-Saclay for his help to organize our workshop. At IDMIT, I thank my colleague from the different facility, Francis Relouza for the Animal Science and Welfare facility, and Sophie Galouette for the cytometry facility, because we are using mass cytometry, also called CyTOF. Nathalie Bosquet from the Infection and Immunity facility for the Luminex analysis, and Paul Mazet from the Bioinformatics facility to analyze both CyTOF and transcriptomic data. The RNA-seq for the macaque cells was done by Hakim Hosseini. So do not hesitate to contact one of us or both of us together. And please join us at our workshop. You can register through the EUGLOHRIA and EUGLO website. And thank again, and I'm happy to take questions.
Thank you so much, Anne-Sophie. We are checking the public and the chat online to see if there's any questions. We can take one question since we're out of time and to not overlap with the next sessions. But it seems it was very clear, your presentation. So we're maybe ask you all, thank you again for your presentation, and ask everyone to join us at the coffee break, the in-person, and at home, please come back again at 11:30 so we can continue these sessions of sharing best practices. Thank you so much.
I see Johanna in the room and Eric and Sonia. Thank you.
Welcome again. So we're going to continue with the plenary session on best practice and lessons learned in shared use of research infrastructure in global health. So the first presentation will be done by Alicia Zamborlini. She's a PI of the CREX HIV project. Whenever you're ready, Alicia, please take over.
Mode, so I think it should work. Can you confirm,
yes, you need to maximize your... Yes, it's perfect. Thank you.
Okay, thanks so much. So yes, thanks a lot for the introduction. This corrects for HIV. I'm not very good in finding acronyms and nicknames, but it means combining CRISPR-Cas9 technology and toxil explants to investigate HIV host interaction and the early immune response towards infection. Myself, I'm glad to be there virtually. I'm professor in virology at the Faculty of Science of Paris-Saclay University since 2019. And in 2021, I joined the CEA IDMIT department as a group leader. I'm very interested in studying molecular aspects of virus host interactions. And I've always been using HIV as a model. More recently, I'm addressing the interplay with particular emphasis with non-second immune cells, such as macrophages, that are target cells for the virus in vivo. I'm interested in understanding how the early immune response is controlled by post-translational modifications, which are reversible and dynamic modification of proteins that can ensure very fast and dynamic response. I'm particularly interested in phosphorylation and SUMylation, and the idea would be to establish means to manipulate this modification and therefore protein functions. On a more technical point of view, I'm also very keen in implementing the CRISPR-Cas9 technology in my studies, and particularly the use of integrative or non-integrative lentivector or ribonucleoprotein platforms. So my collaborator is Joachim S. which unfortunately I'm sorry, don't pronounce properly. He's a senior lecturer and associate professor in virology since 2020 at Lund University in Sweden, and he's head of System Virology Group. Himself and his group have expertise in bioinformatics, omics, and sequence analysis, and they share with me research interest on virus evolution and disentangling how early virus-host interaction are associated with variability in disease progression. He has developed a secondary lymphoid tissue model to study the association between the innate virus-host interaction and tissue pathogenesis during the establishment of the infection. So based on that, we actually connected recently because Joachim is deeply involved in EUGLOHRIA Alliance and was a co-organizer of a workshop about a year ago. So after that, we connected and started to discuss about this project, which again has its roots in our interest in studying the human immunodeficiency viruses. So there are two human viruses that are one and HIV2 that circulate in the human population. As you might know, they are the results of independent zoonotic transmission of simian viruses that infect chimpanzees or Sooty Mangabe to humans. Obviously, simian immunodeficiency viruses are highly spread in simian population in Africa. So HIV-1 is a very, let's say, efficient virus because is the responsible of the worldwide pandemic, infecting about 37 million people, characterized by a very fast, let's say, fast pathological evolution and high transmission rate. This infectious disease, if untreated, is very deadly, leading to the AIDS syndrome in 98% and even more than 98% of people in absence of treatment. This virus poorly replicates in macrophages and other non-cycling immune cells. The HIV-2 virus, although genetically similar and having this similar evolution history, is mainly concentrated in West Africa, infecting between 1 and 2 million people. It has a different physiopathological evolution because it's transmitted less efficiently and leads to AIDS in longer in a lower proportion of people, estimated to be about 25%. And usually time for this progression to AIDS is longer, over 10 years. And this virus at a cellular level replicates more efficiently in non-cycling immune cells. A reason for this difference at the cellular level is attributed partially, at least in part, to the fact that the HIV-2 virus and the original viruses harbor a protein that is called VPX that is absent in HIV-1. And this, the product of the VPX gene, actually, which is the VPX protein, has a very important function in overcoming the activity of antiviral proteins, which has it's proposed to have implication for the immune response. So the work of Joachim previously has shown that HIV-2 seem to have the ability to inhibit the more aggressive HIV-1 virus in context of co-infection in vivo. So based on that, we established a project, we started to discuss and we, the objective of the project that we submitted to the EUGLO call was to set up innovative experimental approaches to investigate interactions between HIV-1 and HIV-2 in their host cell. And of course, to try to understand what was the implication for the early immune response. So our project is organized in two work packages. One that is the experimental part, where we aim to establish the 3D cell culture model using explants that will be used to compare the outcome of HIV-1 and or HIV-2 infection or the co-infection. The second aim is to optimize a non-integrative lenti-vector system to deliver Cas9 and RNA cassettes using initially THP1 cells that are well-accepted surrogate for macrophages, and of course bringing this further then in primary cells and also in the tonsil explants, where we want to study the expression of Cas9 and of course the editing efficiency of the genome of interest before being able to do infections there. The third aim is to assess the consequence of inhibiting post-translational modification, including SUMOylation, for HIV-1 infection and immune activation in the tonsil explant. The second work package also of this project is to organize meetings between our groups. And I'm sorry, I didn't take pictures for this presentation. It's going to be for the next one. So we already, so it's a baby program because, again, we actually never met in person and we connected pretty recently. We started in September, and we had a virtual meeting mid-October 2023, where people working, my collaborators and Joachim's one, presented the results so that we can know each other. So we plan to have one meeting every 3 months, depending, of course, on our agenda. And we also plan to visit each other. So I foresee to go to Sweden in February, and then Joachim will come and visit Paris-Saclay next year in May. So we have a little of results, preliminary results to share with you. So I'm trying to struggle with my screen. So as you know, the CRISPR-Cas9 technology, while it's emerging as a tool that is a molecular tool that is well, cutting edge and widespread in scientific research today. It relies on the fact that the Cas9 nuclease associated with a specific guide RNA can target the gene of interest that will be cut by the nuclease activity and then repaired, leading to the compromising the open reading frame and therefore impairing gene expression. So we will use this technology in two ways. On one hand, we will use the RNP platform that uses the Cas9 protein that is purified, combined with 3 different guide RNA, and this will be used to electroporate THP1 cell line that we developed in the lab to express GFP, to have a fast way to look at the editing efficiency, in the idea to optimize that, because then we can look at the cells that become negative by flow cytometry. In parallel, we also use a lentivector platform, where we have a vector that encodes Cas9 and the guide RNA that will be packaged using the packaging vector and an envelope vector into VLPs that will be used to transduce the same TF1 GFP cell lines, which will be, again, analyzed by flow cytometry. In parallel, we also have set up in the lab the digestion with the endonuclease that allows fast screening of course, a population where the gene that will be knocked out is not GFP, and that cannot be analyzed by flow cytometry. And then, of course, we will, the samples will be used to do sequencing. So for the RNP platform, we did some tests already, where we have the GFP positive cells that are electroporated using different conditions in terms of voltage of the impulse, duration, and number of impulse. And we follow after the electroporation, the amount of cells, which is the bar. We electroporate 200,000 cells. And then we can see that over time, and I'm sorry, this is 3, 5, and 8 days post-electroporation, we can see that there is some recovery of the cells. And at the same time, we can monitor the GFP expression that, of course, goes down below 95% in most conditions. And the idea here is to be able to identify the best condition for cell electroporation. Of course, this approach is well suited to cells that can be electroporated, therefore can be put in suspension, and not perfectly suited for, of course, explants. In this, with this idea in mind, we also use this VLP platform, where we produce VLPs harboring Cas9 and the guide RNA that are either non-integrative or can integrate. And as you can see here, cells, the THP1 GFP cells were transduced, and then we monitor the decrease of GFP expression, and we can see that at day 8, the two platform seems to be very efficient. But this is preliminary data, and we're still working on that. In parallel, we set up this endonuclease digestion assay, where we amplify the region that is of the gene, in this case GFP, that is targeted by the guide RNA. And then the PCR product is incubated with an endonuclease that will cut heteroduplexes that are formed because I forgot to tell you that the cells, the samples, they are heated, and then we cool them down so that the PCR products, they can form this heteroduplex. And as you can see here, there was no digestion for the parental cells where there is no heteroduplex, while the cells that were treated with integrative VLP display this panel with different bands that indicates that indeed editing efficiency, editing occurred. And we can use the relative intensities of the band to calculate the editing efficiency. So again, these are data that we obtain after the summer break. In the meanwhile, Lund University set up a pipeline to obtain toxin that will be used to perform the experiments. So they have a a well-established collaboration with a hospital in south of Sweden that collects tonsils from healthy donors after informed consent. Then the tonsils are shipped to Lund, where they get, as you can see here, dissected, and then small slides are generated, put in culture, where therefore the toxin explant can be treated by the CRISPR-Cas9 technology for editing the genes of interest, and then infected with HIV-1 or HIV-2. And after suited time, the samples will be hybridized and analyzed using different approaches, including transcriptomic, spatial transcriptomics. What is our feedback about the added value of EUGLOHRIA, and what are our experiences? Well, like my colleagues said before, we have a very positive feedback on the fact that, of course, the EUGLO Alliance has created for us the opportunity to establish a new collaboration among our research group in Lund and Paris-Saclay, based on the fact that we have common scientific goals, but we use in our experiment complementary approaches. So we really hope that we will benefit from synergies that will emerge through regular scientific discussions in common seminars and meetings. Of course, we expect that we will acquire new skills and generate also preliminary data that we will use to apply to common grants from external fundings, national agency in France and Sweden, or European funding agency, where like the other colleagues said, we are discussing about it. We also think that this collaboration can increase our visibility and the impact by using through the writing common publications, and of course giving access to a wider panel of infrastructure and platform that are run by expert staff, and therefore makes it possible to ask novel, more ambitious questions that we would ask independently. I should say that listening to my colleagues before, I would like also to stress the fact that, this collaboration, really hope that it will be long-lasting, and we are really committed to that. But definitely we will need support. One point that I can bring up is for sure the fact that it's going to be high beneficial for us to have co-direction of PhD students. And maybe the other thing that I can say is that, to my understanding, both me and Joachim have academic positions. So we have heavy teaching duties, and of course, we could also benefit if having more time for research is if these teaching duties were a little bit lighter. That said, well, of course, we received, as our colleague, funding, and we will write a report on that. So some of the fundings will be used for travels. Fundings have been used for buying reagents to perform the experiments, and also for the platform on our side. And with this, I hope I was not too long. Thanks for your attention, and of course for the support we had from EUGLO staff, particularly on my side, Eric Cassan, that is in the room. So thanks a lot.
Thank you so much, Alicia. We're just checking the room and the chat to see if there's any questions for you. Okay, one question. We're going to pass on the microphone.
Hello, Alicia. Eric Cassan, évidemment. Of course. So thanks a lot for the presentation and for the acknowledgements also. This is not a question, but more a remark. What you have introduced, what you have explained to us is the fact that you have never met Joachim Ersbjørnsom. In fact, physically, I mean. Okay. And this is an outcome of EUGLOHRIA. It shows that research collaborations can be initiated remotely, and after they will be supported by missions, etc. There is also, and this is a reminder for everyone, a conference organized in Munich, in München, at early of December, from 4 to 6 December, and the PIs of the different projects are invited to meet each other in Munich. And for you, it may be the first opportunity to meet even before February, because I have understood that you will travel to Lund in February. It would be an opportunity to meet physically. I cannot comment the scientific content of your presentation. What I can see that I can share with some colleagues in the room. There are some vice rectors, some academic leaders, etc. I think that our universities also have to help the PIs, the researchers, to apply to competitive course. I mean that we have also, yesterday we have at the round table, Sonia Pereira from Porto, who is in charge of the head of the department for research projects, etc. We have similar departments in our university. I think that within EUGLO also we have to help the researchers not only to get directly fundings, but that fundings, the EUGLO label fundings, may be always a bit weak. It can enable missions, etc. But beyond that, we need to mobilize our departments for writing and helping the researchers to write and to draft a research project to be submitted to competitive course. Okay, thanks again, Alicia. See you soon.
Yeah, if I can stress that. Thanks so much, Eric. Yeah, I think that the support from staff involved in the EUGLOHRIA Alliance is key. It's very important. And so, yeah, please help us doing that and go through these very competitive European calls, because I agree with what you said earlier, that we have to think about cost actions, PhD networks, the other point I wanted to make is that indeed I'm, listening to colleagues, I learned that there are opportunities, of course, for travel and, these joint PhD programs. So yeah, I hope I will meet some of you in Munich in December, indeed. Thanks so much.
Thank you so much. We're going to move on to the next presentation. We call to the stage Christina Bozaz and Mahia Ahmati for the presentation of the eVine project. Thank you so much.
So, my name is Christina Bozás, and I work in University Szeged, and we start the collaboration with Marianne Debly, who cannot be here today. So I try to present you our motivation, what was the what initiate the to start this collaboration, and my excellent postdoc tried to solve the this task that she will present you Professor Debly's slides and show you our preliminary results. So what was the challenge? The challenge is the spreading of cancer. So the number of people newly diagnosed with cancer every year in Europe is more than 3 million, and this number is growing. And I think all families touched with cancer, you and or your brother or sister and friends and parents could suffer from this horrible disease. And we know that the success of therapy and the chances of survival depend, among others, on the early diagnosis. But the people doesn't like go to the doctor. They are afraid of the procedures required for diagnosis, and especially afraid of the result of the screening. But what will happen if we can solve this problem with a simple blood test? So the biomarker research tried to give the answer for this question. You know that the biomarkers are biological molecules found in any kind of body fluids. And the scientists try to find one single molecule, which can describe the pathophysiological status of the patient. So this is the conventional way to find the biomarker in the body fluids. But this way has many limitations. The conventional methods to screen the body fluids, the proteomics, lipidomics, genomics, and the transcriptomics. And these methods are so expensive, the time-consuming, require great expertise. But my opinion that the human body is too complicated to describe the pathophysiological status with one molecule. So we use the other way to answer this question, namely the extracellular vesicle research. The extracellular vesicles are nanometer-sized, small lipid-bounded bodies in all body fluids. And these lipid balls stuffed with many kinds of molecules, with proteins, hormone-like molecules, or different amino acid molecules. And we can screen these small bodies, the extracellular vesicles. And we have novel approximation in this type of research, because we try to use the molecular spectroscopy methods, which give us a specific spectral pattern. So we use the Raman spectroscopy to screen these small bubbles with the Raman spectroscopy. I will not give a lecture about the Raman spectroscopy because I'm not brave enough, because Professor Konya is sitting in the audience. But the essence of this method, that the Raman spectroscopy is a vibration spectroscopy, and we gain the information on the total molecular content of extracellular vesicles. So our objective was to develop a generalized Raman spectrum-based classification algorithm that can support diagnosis or prognosis in clinical practice. And we have a special approximation that the unit of the information is the extracellular vesicle, not a single molecule. We formed different patient groups. The control groups was the lumbar hernia groups, so there was the non-tumoral groups. And we have two really aggressive brain tumors, the glioblastoma multiforme and the brain metastasis from the lung cancer. The not so malignant brain tumor, the meningioma, the melanoma patients, and rectal carcinoma patients. So now we have almost 500 Raman spectra, and now in this moment we analyzed almost 300 of this. The method, the workflow was so simple because we are obtaining plasma samples. After this, we purified extracellular vesicles, and we completed the Raman spectra. After these Raman spectra were analyzed by machine learning methods and neural network analysis. And we have found that the control groups versus brain tumor types can divided with about 80% specificity from the control. So this high percentage could be useful, for example, to follow the success of the therapy in the metastatic disease, or it could serve a good method to diagnose the brain tumor without any expensive methods such as the MR or the, for example, the PET-CT. The platforms that we use, the Surface Science Center for Raman spectroscopy and the Biobank of University of Szeged. We have many collaborations with clinics in Szeged. And so this was the background which motivated us to make the collaboration with Marianne Deblis at the University Paris-Saclay, because we tried to find the alternative diagnostic and prognostic opportunities based on extracellular vesicle communication and the three-dimensional brain tumor models. So this is my sweet, young, and pretty research group. And Maria will continue my presentation. I would like to thank you for your attention with my own flowers in my garden. And let's see, there is a sweet ladybird. So Maria is coming now.
Thank you, Christina. Okay. I can see my slides, but not over there. So, as you heard, I will talk about this EUGLOHRIA research project. I will replace Marianne Deblis today. This project is on the comparison of extracellular vesicles in low and high invasive diffuse midline glioma. Okay, so our research partner is located in the Gustave Roussy Institute, which is dedicated to cancer, and they have an integrated research program related to pediatric brain tumors, mainly in diffuse intrinsic glioma. And they have an access to clinical samples and also clinical and radiological data. And so they develop different kind of models of the disease, such as 2D and 3D cultures, and also orthotropic xenografts. From all of these samples, they perform genomic profiling, including epigenomic profiling, because the pediatric brain tumors are related to epigenetic dysregulation. This has a significance. And from all of these models, they also perform drug screenings to find new therapeutic targets. And Christina talked about our work. So, as you know, we work on tumor-derived extracellular vesicles in different kind of basic research projects and clinical studies as well. In this EUGLOHRIA project, we focuses on the diffuse midline glioma, K27 altered, which is a rare disease, but it is still one of the biggest challenge in pediatric oncology, because the tumor cells infiltrate the surrounding cerebral tissue, and this infiltrative nature makes this tumor non-resectable, and this tumor leads to a very low median survival, unfortunately. And molecularly, this tumor is characterized by a point mutation in the 2 genes of histone H3. And this leads to a dominant negative inhibition of this mutation. This is a repressive epigenetic mark. Okay. The team of Marianne Deblis also developed an assay for investigation of the invasion capacity of diffuse midline glioma cells, and they can quantitatively describe the invasion capacity of the tumor cells using 3D in vitro tumoroids. They insert the tumoroids into matrigel to mimic the extracellular matrix. Then they perform the quantitative analysis, and the results of this invasion assay is strongly correlated with the metastatic progression of the disease in the patients. They also investigated the morphological and molecular characteristics of these glioma cells, and they found that the low invasive cells have a mesenchymal morphology, which relies on strong focal adhesion, and the high invasive cells has amoeboid morphology. And in this EUGLOHRIA project, we aimed to investigate whether the tumor-derived extracellular vesicles can reflect the invasion capacity of their tumor cells. So we aim to isolate the extracellular vesicles and investigate their molecular fingerprint. We have chosen 5 low-invasive cells and 4 high-invasive cells, isolated their extracellular vesicles using size exclusion chromatography, and investigated them by Raman spectroscopy to analyze their molecular fingerprint, and we plan to use proteomic analysis as well. So the first result, we measured the particle count of the tumoroid samples before and after EV isolation as well. And as you can see, before EV isolation, the particle counts was similar in the low and high invasive tumoroid groups. And during the EV isolation, the particle counts decreased because we only retrieved the small extracellular vesicle population. And interestingly, we found that the low invasive glioma tumoroids released higher amount of vesicles than the high invasive group. Then we measured 5 parallel spectra of each vesicular sample, and also 2 control media as a background, and we performed baseline correction and normalization on these spectra. Then to reveal which samples are similar to each other based on their spectral fingerprint, we used the so-called TSNA plot, which represent the data series with multiple dimensions, only in 2 dimensions. So together with hierarchical clustering, our analysis showed that high invasive EV samples are similar to each other, they are grouped together, and the low invasive samples are separated from them except 1 sample. So in conclusion, we found vesicular differences between the low and high invasive tumoroids, and we have promising results, but we want to increase the number of samples and also the measurements to use classical machine learning models for the analysis of the Raman spectra. And we think that these results may provide the basis to extend these analyses to clinical data, to clinical samples, namely blood vesicles in diffuse midline glioma. And €10,000 budget in the last 3 months was spent for the experiments and also laboratory visits. And we used a research platform, the Surface Science Center at the University of Szeged in the laboratory of Professor Zoltán Kónya for the Raman spectroscopy. And I think that both of the research groups benefited from this EUGLOHRIA project so far. So the French partner provided the expertise on diffuse midline glioma and the valuable clinical samples, tumoroids, and the invasion assay, and the Hungarian partner contributed with the 15 years experience on tumor extracellular vesicles and provided the research platform for Raman spectroscopy. And together we obtained promising results, and we believe that this will help to find further grants to extend our analysis to clinical samples to reveal the prognostic value of extracellular vesicles in diffuse midline glioma. Thank you for your attention.
Thank you very much, Maria. We're scanning the room to see if there's any questions. Seems there's no questions, so we're going to move forward to the next presentation. The next presentation is from Nabila Sedeki. She's a PI from the Neonate project. Nabila, whenever you're ready, please take over.
Thank you so much. Can you hear me properly?
Yes, we can hear you fine.
Okay, thank you. Just sharing my screen. Hopefully it will work fine. Can you see it?
Yes, if you go ahead and put it in presenter view.
Yeah, absolutely. Let me just... It's fine from your side?
So we are seeing another window. If you can share your other screen, that'll be great.
Is it better now?
Yes, we're seeing your slides, but it's not on presenter view. Could you maybe click on F5?
Is it better now?
We're not seeing it on presenter view, but I think you can move on, and maybe if you switch the slides, we can follow your presentation.
I'm so sorry. I can't see it properly from my side, so it's like Anne-Sophie, I'm not used to Zoom. We work with Teams usually, so okay.
Can you share your screen again? I'm sorry, we're not seeing. Okay. Can you click at the top of your PowerPoint presentation, fourth button, so you can begin the presenter view. Or if you prefer, you can go ahead and start your presentation like this and switch the slides. Hear us? Nabila, can you hear us?
Yeah, I can hear you, but I don't know how to make it better. Sorry.
That's okay. You can go ahead and start like this and switch to the slides.
Okay, all right. So can you see something at least?
Yes, we see our presentation.
Okay, so sorry about that. So thanks for invitation. So on behalf of my collaborator, Claudia Nussbaum from Munich, I would like to present you our very recent new collaboration in the context of the EUGLOHRIA network. So we would like to understand the mechanisms underlying the infections, RSV infections specifically in early life. So who are we? So I'm an immunologist working at the CEA, IDMIT, which is part of the University of Paris-Saclay, together with Claudia Nussbaum, who is a pediatrician, scientist, doing her research at the Ludwig Maximilian University in Munich, LMU. We met basically on Visio, we haven't met again in person, but we are really keen to do it in the next month, hopefully. And so we are very interested in working together and sharing expertise and knowledge towards improving neonate or children health. So at IDMIT, as Anne-Sophie highlighted, we use the non-human primate model, the preclinical model, to understand how the immune system works towards modulating how the interaction between the immune system and the pathogens work. And we work on several different pathogens, which are listed here. And of course, we use the adult preclinical model, but
Sorry, Nabila. Sorry, Nabila. We're seeing the first slide. I'm not sure if you're switching slides.
Yeah, I am actually. But
maybe you need to
seeing...
share your screen, your first screen. Yes, now they are moving. Okay. That's great. Thank you.
Yeah, okay. It's not in full screen though. No?
That's right. Yes, it's not on full screen.
I mean, let's do it this way. Sorry, but yeah.
Thank you.
Okay. And sorry again. I was explaining that at IDMIT, which is the infectious diseases for model to understand the infections, the infection in human. We use the preclinical model towards understanding what's happening in human, of course. And as I said, we use all these different, or we are interested in all these different pathogens. And in the context of the collaboration and with Claudia Nussbaum from LMU, we are interested in neonatal, in early life infection. And at IDMIT, we developed recently the PIP program, which is the Pediatric Infection and Immunity Program, which has been implemented in late 2020. So our aim is really to understand how the immune system develops and matures in early life, and how this immature immune system can cope with the different infections. So with Claudia at the LMU, we are thinking together to put our expertise and to move forward towards improving or understanding the immune system development and the infections. And when I joined the EUGLOHRIA network, basically, it was very helpful because that's how I found my collaborator, Claudia, who actually she is involved in this new network, which has been implemented very recently. And this is a large network which is in Germany. It's a German network which englobes all these different part of Germany. And it's very fortunate because it's exactly they are interested in the topics that we're interested in here at IDMIT, going from the intrauterine life through the perinatal changes towards the postnatal life. So the aim of the pilot network or consortium is to explore the mechanisms that determine the perinatal differentiation of immune cells and their cellular environment. So this is really very fortunate because it's exactly what we are really keen to develop here at IDMIT, using the preclinical non-human primate, which is really the model that can replicate the human disease. So the project in the general objectives of the project is really to study the mechanisms underlying immune responses to viral infection that affect neonates and young infants, to anticipate adult infections and by this way improve human health. So we will use the preclinical NHP model, as I said, which replicates human infections. And this project will build a solid platform for further respiratory threats. So more specifically, the context of the collaboration with Claudia at LMU, we have specific objectives. So we would like to characterize the immune responses to RSV, the respiratory syncytial virus. Why we are interested in this virus? So as you can in this see in this small graph here, you can see in blue the hospitalizations basically through 2022 and 2023. Basically, this large peak of infection or hospitalization is really towards and focused on the neonates from 0 to 6 months, from birth to 6 months. So we can see that the RSV is still a real burden, and we really need to do something about it. Of course, currently there are some new vaccines and new therapies to help addressing this public health problem, but I think I'm not talking about this today. Hopefully, we will meet soon to talk maybe further on this. But we feel, and the community, basically, research community, feels that we need to better understand what's happening exactly in early life when this immature immune system encounter this virus. So we would like to address these 3 specific aims here in this pilot study with the LMU team. So we would like to infect cells using in vitro to set up a system where we can understand and study the interactions between the RSV, the virus, and the immune cells. Would like to characterize the phenotype and the function of these cells in the presence or not of the virus, and would like to compare these parameters between neonate and adult cells, and understand why the RSV is making really is complicating this infection in neonates, but not in adults. But of course, we know that in elderly, it's another problem, but we are focused on the neonate first. So our first results, basically, it's just really to start discussing about this with you. This is the frame of our project. We managed to set up an in vitro system where we take the cells from the bronchoalveolage lavage from the non-human primate, neonate non-human primates. And of course, in parallel, we do adults. And we took these cells and put them in the presence of the virus. So this is a virus that has been used for the challenge in human, a study that has been published in 2020. And this is a collaboration we do as well with the Imperial College in the UK. We got the virus and we managed basically to infect the primary cells. And this is up here. We can see that the virus, this is the mock. Basically, we don't see any infection. And you can trust me because I know the pictures are too tiny. But we managed basically to infect and to show that the virus gets into the cells from the macaques. Here in the lower panel, we can see that the cells basically change phenotype, and you can see that they are more activated when the virus is in contact. So the next step is what we are currently doing is really we built the panels to be able to look at the lymphoid and myeloid cells in this co-culture of BAL cells with the virus, with or without the virus. We started building the panels. We bought the antibodies and we did the primary or the first experiment just to see if everything was working well. And indeed, we managed to get the panels, the two panels working. And we are currently discussing with the LMU team to see how we can mature more this and go and of course apply these panels to cells from human neonates provided by our collaborators at LMU. So we are interested in specific populations, of course the macrophages, because it's the dominant population in the BALs. And of course, we are interested in neutrophils and B lymphocytes because they have been shown to have an important role in the RSV infection. So what about the budget? Because we were asked to talk about this. So we just received the funding. So thank you so much for the EUGLOHRIA. And ordering are in place. We already bought the antibodies, the tools that we are using for the cytokine and chemokines in these co-cultures. We are, of course, discussing a lot with our collaborators and platforms. So the experiences from the perspective of from both LMU and IDMIT CEA and Université Paris-Saclay, basically thanks to the EUGLOHRIA network, we were able actually to meet and network with Claudia and her team. Meetings are ongoing. We had already 3 meetings. So this is, as I said, a very recent collaboration. We haven't met yet in person, but we collaborate, we discuss, and we think that thanks to this collaboration, we will be able really to meet the pilots network, the German pilot network, to really extend our collaboration. So the aim is really to use this platform and to go now and to ask for more funding at Europe. Hopefully, EUGLOHRIA will help again with the funding, and we are, of course, thinking to apply at more national fundings from both ends. The added value, so indeed, this is very rich, because we could really share now knowledge and started to share expertise. The platforms are key, of course. From our side, we have the platform of what we call L2I, headed by Nathalie Bosquet. Thanks to this platform, we will be able to look and to measure the cytokines and chemokine receptors within this in vitro system. Thanks to the flow cytometry platform headed by Anne-Sophie Galouette, we'll be able to determine the phenotype and the function of the key players in the RSV infections. Of course, the ASW, which is the Animal Science and Welfare here platform at IDMIT, it's capital, of course, to have access to the cells, the BAS samples from the non-human primate preclinical model. And of course, from the research team, we have the pediatric research team here at IDMIT, and together with Claudia's team, we will join expertise and knowledge to be able to answer to these key questions. The added value, so we've been pretty productive, because even before meeting, we had the opportunity, and thanks to Eric Cassan, who was key actually to really make us network with Claudia, we were able to organize a workshop on the 5th of June in 2023. Basically, it was really productive. We had a very nice program, and we had young investigators as well from the EUGLOHRIA network, which was really interesting. We talked, of course, because the main topic was RSV, because it's a real burden. We need to talk about RSV, but we talked about other topics in the context of EUGLOHRIA. We had young investigators and senior speakers. It was really rich. The added value was, of course, this collaboration. We, as I said, we will be able to collaborate with the pilot network in Germany, which has been funded recently, and I think we can have access to funding to meet in person with Claudia, and of course, thanks to the support of EUGLOHRIA. So we are very keen to have more support, and thank you to everyone, and hopefully we are keen to meet in Munich, hopefully in person, in December 2023. And again, sorry for the slide issues we are having. We still, I think, but hopefully you could hear me properly. And I'm really keen to answer questions if you have any. Thank you.
Thank you so much, Nabila. So we're scanning the room and also seeing the online chat to see if there's any questions. I believe there's no questions, so thank you so much again for your presentation and for bearing the technical difficulties, and also clearly presenting your work and sharing your experience. We're now moving forward with the presentation of Dusan Palik. So he's the PI of the Aquarium project. Whenever you're ready, please take over.
Just give me a second. Hello, everyone. I'm trying to, okay, so we are two persons here on the side of LMU München, myself and our associate on this project, Dr. Konrad Wojnarowski. Let's see how then we can share the screen. I hope everybody can see this, right?
Yes, we're seeing your screen, but it's not on presenter view. So if you can go ahead.
Okay. Let me start this. Is this better now?
So we're seeing it on presenter modes. You need to, I think you're using two screens, right? You need to
Correct,
switch
yes.
the screens.
So we can just then
duplicate
this.
Yes. Okay, that's great. We're seeing it perfectly.
Okay. Great.
Whenever you're ready.
So we're going to be talking about something that may be a little different from your topic of or regular medicine, human medicine, I should say. We are both from veterinary background, and our colleague from University of Porto, she's from the aquaculture background. So we are discussing a possible next health crisis that is emergence of antimicrobial resistance. And it has been deemed as a problem for the last few years. This emerging of antimicrobial resistance was connected to the aquatic ecosystems and providing this information from the aquatic ecosystems that could use or be used as monitoring for additional information that we can get about what's coming down to us from the perspective of antimicrobial resistance and the spread of the resistance in the ecosystems. So the LMU team from the chair of fish diseases and fisheries biology, it's three of us, myself, then Dr. Paulina Kolewinska. She's not there with us today, but Konrad and her are working really on the meat of this proposal. We essentially work in the fields of aquatic veterinary medicine, but we have some expertise in animals' microbiome, One Health, and some endocrine disruptors issues. Our colleagues from University of Porto, maybe you know them, or you're familiar with them. It's Professor Valente and Dr. Monteiro, and they work in fish nutrition and sustainable aquaculture, but as well also in One Health areas. Briefly describe what we want to try to do, or what we're trying to do, is to provide a cheap and quick tools to broader audiences in order to be able to do surveillance or monitoring of the emergence of antimicrobial resistance from the aquaculture systems. Why is this important? I won't go into huge detail, but we are eating more and more fish, and global perspective of the seafood consumption is increasing. I mean, we're globally producing a lot more fish than we did only 10 years ago, and we're satisfying the protein requirement for growing human population with aquaculture, with producing mostly finfish, but other organisms as well, in a controlled environment and the life cycle. And with that, disease risks come in. We have to use treatments, antibiotics included, and we put some pressure on the organisms that are consisting of the micro environment in these animals, aka microbiomes, and pushing them into the direction of possible development of antimicrobial resistance. So our idea was to use a cheap, when I say cheap, I do mean cheap. The mini ion costs is affordable to pretty much almost any bigger farm or veterinary system, because it costs about €1,000 per piece. And together with all the reagents and the cost of this stuff, we're talking less than €10,000 investment into something that could give you fairly quick information if you know how to do it. And we are developing basically a streamlined procedure to get the extraction of the DNA together with long reads and short reads. Most recent information that Conrad is going to talk about, that we actually are focusing more on long reads in this segment, and then do the streamlined bioinformatics analysis using freely available tools that are developing and advancing pretty much every day. So we're trying to give the tools to the farmer, essentially, and to the colleagues who would be on the front lines of the monitoring and existing problems with antimicrobial resistance. So briefly, as I mentioned, the risk of the gene exchange of the antimicrobial resistance can actually be higher in the aquatic environments. And this creates a gap in our knowledge about dynamics of these mechanisms and routes. We simply don't have this information. So we also figured out that there is a missing link between the big centers for sequencing and then easily affordable technology to provide this lack of standardized sequencing protocols and bioinformatic flows. And if we are able to streamline those, we might be able to monitor the large number of samples which is needed for a detailed surveillance of everything that's going on around us in the aquatic environment. So we have a hypothesis to look into that microbiomes actually in farmed fish can be utilized in a surveillance program to detect and monitor emergence of genetic patterns that are favorable to development and transfer of MR genes. And our objective of this pilot project is to integrate low-cost, novel next-generation sequencing platforms with streamlined bioinformatics stuff to be able to analyze microbiome samples from farm fish with and without exposure to antibiotics. And of course, we cannot do this directly, and in the large scale, we have to do some initial adjustments first. That's why we're using samples that we collected from our original experiments from different projects, both in the area of our colleagues in Portugal, who are really into functional nutrition and animal welfare. So they are working on screening functional ingredients to promote better health of the animals, of the fish in this case. They are increasing or enhancing host responses to infections because they improve gut health. And when they do the challenge studies, they see if the diets, the functional diets, work this way or the other. But those functional diets also change the microbiomes in the gut. And that's where we come in with collecting the samples. And I will turn this to Conrad for a few minutes.
So as Professor mentioned, our approach will be standardized and streamlined to exactly fit the needs of the project, and it will allow a replication in the future by actually nearly anyone. The results that we were able to achieve with this approach are, for example, the analysis of the microbiome coming from the fish intestines. We were able to see and notice the differences in different groups of animals, fish. With this kind of similar approach, we're also able to find antimicrobial resistant genes coming from direct culture of Rhamnosus salmonicida. We found a number of those, and it's only the beginning. The same approach can be also used for checking more, I would say, complex samples. For example, here, in this case, we were able to find antibiotic resistance genes in microbiome that was on the surface actually of the liver, in the liver of the fish. Some of those, as you see, were present there. For example, MF2, PSA or FPV54, which are prevalent antibiotic resistance genes. Those complex samples that I mentioned before were, for example, sediment from the river. And this part is one of the most interesting things, because at the same time we were checking microbiome composition, And with that same actually analysis, we're able to find antibiotic resistant genes present in the sample. So tied to this sediment sample were antibiotic resistance genes like TAM116 or BEL-A TAM116, to name some out of the many. Also, when we checked the water, also like the developed microbiome was to be seen, but behind it, actually we found another group of antibiotic resistance genes that were present in microbiome that was, I would say, kind of connected to the samples that we had and was living in it. Back to the professor.
So with this kind of a brief and a quick summary on what we have been doing, we have found that there are definite options for further collaborations between our two groups. As was mentioned by the previous colleague, we didn't get a chance to meet in person yet, but we're hoping to be achieving that fairly quickly. So one of the things that is beneficial from the aspects of the EUGLO, the whole action, is that and due to some of our connections with different committees at the level of the tripartite commission, the commission that is dealing with antimicrobial resistance at the global level, from World Health Organization, World Organization of Animal Health, and FAO, the Food and Agriculture Organization of United Nations, we may be able to access them, and actually with the results of this quick pilot project, ask for some additional funding to train the trainers in this streamlined approach. With the experiences, we are really happy about the quick and rapid responses and communication from our side as investigators and the side of the colleagues at the administrative aspect of EUGLOHRIA. I must say that when first EUGLO project started back just before Corona, basically, I think it was the time, we were approached, or Professor Valente approached me to give some initial lectures on One Health within this platform. And what we did was that we started the collaboration back then, but then it was halted, as pretty much anything else was in that period of pandemics. And this kind of, I will say prepared us, but we were kind of in the loop of communication. And when the opportunity presented from the LMU side to get some of these pilot grants, we took it, and we were really happy about the simplicity and the quickness of the process. However, this has also a bit of a negative side. The highest limitation that we figured out, we have a very short lifespan of this project. So basically, we were informed that we have the money fairly quickly after the application was submitted, but then we were told also, and this was already kind of announced in the conditions, that we have to finish this by December. And this doesn't leave, if anything, trying to prepare the animal experiment or collect the samples from de novo from the situations gets to be at least a year-long effort in many cases. So in our particular case, what we then focused on is our database of samples already collected from previous projects, in order to be able to additionally do some sequencing and analysis, to be able to use the collection that we have in order to put together a meaningful story at the end. Another example for that is we knew this since about July that we get the money. So even with, from July to December, if we pay attention to our busy schedules and semester and teaching and everything, it is very difficult to organize visits that would fit in everyone's schedules and additional other work. So those are comments or experiences with EUGLOHRIA, and definitely there is an added value to this collaboration between University of Porto and LMU, because the CMAR, the platform of CMAR is involved, or CMAR is involved in the EMBRC platform for testing the functional diets at much larger scale that we can do here in the LMU. So we are very happy to receive those samples collected by the colleagues in Portugal. As far as the added value, this is really the first step, allowing a closer connection between our two groups. Since University of Porto has a certain focus on those functional diets, promoting fish health and welfare, while we are working in the area of aquatic animal diseases and veterinary medicine from the perspectives of biosecurity and surveillance and we can combine these two expertise very nicely through this EUGLOHRIA aspects. So this is an added value for us, for sure. And with this, I thank you, and we're open for the questions.
Thank you so much, Dusan. We're scanning the room to see. We have time for a few questions, if that's okay on your side.
Sure.
It seems we don't have any questions, so thank you again for your presentation. It was very nice to have you here. We are now taking a lunch break for the conference. We'll be back at 2 o'clock. So we invite everyone in person to join us for the lunch break and networking part. Thank you so much again, and see you soon.
Bye-bye. Bye.